Okay, everybody, good morning. This is the third part of this little lecture on enzymes for Chapter 3. If you do have guided notes, this is the third video, and you'll be on the back of that packet I gave you. There aren't many blanks and things to fill in on these guided notes, so really be efficient with the amount of space you use, and just kind of keep things pretty condensed. Okay, so you should know by now that enzymes are proteins and they have a 3D shape, and different things can affect that. Okay, and we usually, when we say that, we mean affect their efficiency. Okay, so enzymes can be distorted, this 3D shape, by temperature. As you know, particles move faster as temperature increases, so the enzyme can start shaking so much that its efficiency is affected. pH, extreme pHs can tear enzymes apart and bond with things that would decrease the effectiveness of the enzyme. And then just very broadly, chemicals. There are things that can sort of block the activation site of enzymes, and there are things that will compete for that spot. Almost like when you're trying to get the front seat of the car and you run out and yell shotgun, there's competition to get in the enzyme's active site. So maybe the thing that doesn't belong there tries to jump in that seat. So anything that distorts the shape distorts the function, or I should say changes the function. So enzymes are really Goldilocks. They like to have everything just right, and just right is usually the conditions they evolved in. If you go take one of these bacteria that live in these, hot springs out in Yellowstone, their enzymes will work in those extreme temperatures. Which, by the way, I just saw on the news that a couple guys were arrested for trying to cook a chicken in the Yellowstone hot spring. I don't think that's a really good idea. That's a very delicate environment that you could really screw up. So they don't like you to do that kind of thing. And that's why they arrest you when you do it. All right. Anyway, sidebar. So, like I just said, optimal conditions. I called that Goldilocks, okay? Goldilocks didn't like her porridge too hot, too cold. It had to be just right. And if we introduce pH into the Goldilocks and the bear story, maybe that would also be the case. So for optimal functions, they have optimal conditions. So the rate of enzyme activity will increase with temperature. So when particles start moving faster, there's more particles bumping into each other, but after a certain point, the enzyme will just break down and the thing won't fit at all anymore. So usually we like high temperatures for reactions, but after, like I said, there's a point where it just can't work anymore. Okay. Enzymes work best at specific Goldilocks pHs, okay? Anything outside of that can cause the hydrogen bonds in the enzyme to break, and that's going to definitely change its shape. And once it changes its shape and function, they just don't work too well together. So I'm going to show you a little graph that'll probably start becoming familiar to you. Okay, you should be able to interpret graphs like this. Got three different enzymes. Got the one with the black, the red, and the purple. You can take them, these are temperatures, okay, so temperature goes across the X here. And rate of reaction, how fast things are going to happen. Things happen for this black one here, it looks like a little bit above 20 degrees. For this red one, mid 50s, and for this one here it looks like high 80s. Now you can kind of see here that the enzyme will still work as we get out of the Goldilocks spot, the optimal spot, okay, is the technical term there. But as you get farther away from that spot, the enzyme's efficiency breaks down, and then it reaches a point where it doesn't function at all. So you should understand that you could make the same kind of graph for pH and for competing chemicals or for reactive chemicals, something that's going to react with the enzyme. So, just to review that, enzymes are very Goldilocks. They work best at the conditions they evolved under. Okay, enzymes sometimes have things called cofactors. Cofactors, the key thing here that you probably really ought to know, cofactors are non-proteins that help enzyme functions. Okay, these cofactors are metal-based, and they can bind to enzymes in different ways, okay? A holoenzyme is an enzyme with the cofactor attached. So if you have some enzyme out there that's bonded with its cofactor, we can call it a holoenzyme. So there are cofactors, so this is all factor base here, and then there are coenzymes. Okay, coenzymes are organic cofactors, and vitamins are the most common one. There's certain enzyme reactions that do not happen without essential vitamins that your body has to obtain through nutrition. Usually your body can't make most vitamins without outside help. So these are things that are assistants. The other type of thing you're going to look at, so put both these under cofactors. Make sure you have a heading, something about cofactors. There are also, next we're going to talk about inhibitors. Okay, if I inhibit you from, going out in the hall, that means you're not allowed to go. Inhibit. So make sure you have something about this heading on this section. So inhibitors do exactly what it sounds like. They reduce activity of very specific enzymes. The inhibitor for one enzyme might not have any effect on the other. So this can be really interesting. A lot of poisons or toxins will bond permanently with your enzyme, and then the enzyme's pretty much done. This is like going around putting super glue in all the locks in the school. You're not going to fix that. And that's pretty toxic behavior. Reversible inhibitors, they have weak interactions. All right, this would be like going around putting tape over all the locks in the school. It's a pain in the butt, but we can pull the tape off and still get the key that goes in the lock. So when I use this lock and key idea, the keyhole or the lock is the enzyme and the key is the substrate. So inhibitors could be permanent, putting super glue in the locks, or they could be reversible, putting tape over the locks. You don't want to do either one of those around here because they'll both get you in some trouble. All right. Looking at a cartoon of this here. So you have this enzyme. It's got its active site. This thing that looks like a goldfish cracker. It's going to head into this active site. The enzyme will do something to it. So the competitive inhibitors reduce the enzyme activity by blocking. Okay? So look, the competitive inhibitor doesn't have to be the exact shape. Okay? It can just block one part of the active site. Now, how do you make this problem go away? You put more substrate in there, okay? Let's pretend like you had a bowl of goldfish crackers and you were picking them out and eating them, and I went and put a bunch of blue Skittles in there. Those don't mix very well, okay? How could you make sure your chances of picking out a blue Skittle went down? Dump a whole bunch more goldfish crackers into the bowl. You might get one every once in a while, but in general, if we reduce the amount of blue Skittles or we increase the amount of substrates, it gets easier for this inhibition to be reduced. So make sure you know that the goldfish cracker is the substrate, not the blue Skittle. Okay, you can kind of see there the blue Skittle doesn't let the goldfish swim into the active site. All right. Non-competitive inhibitors. It's a tongue twister for me. They bind to a different spot. The allosteric site. And that changes the shape. And when you change the shape of the enzyme, the desired substrate won't bind properly. Now, some of these are intentional biological controls. Your cells might put out an intentional inhibitor to say, I don't want this enzyme to function right now. Now look very carefully at the blue one on the right. See how its shape is changing here when I advance my cartoon? Look at it right now. Goldfish shaped hole, and when the red thing comes in there, it chemically bends the hole. What's the hole called? Active site is right. And it's not really competing. It doesn't really matter how many goldfish there are. It's just not going to work with the inhibitor in place. It's not competing, it's just turning it off. Allosteric inhibition is what this is sometimes called. So our little fish can't get into his active site. Our substrate goldfish. Okay. So, cells have to always be able to regulate what's going on inside of itself with all these different metabolic pathways. So, how do they do this? We're kind of hinting at it here. Cells control where enzymes are active and when they are active by switching off and on certain genes that code for particular enzymes. Just remember, you have this big set of 23 chromosomes. They have all the codes for all the enzymes you're going to need, but not every cell is making those full time. Your cells only make the enzymes they need when chemical situations that control the cell's environment are set up to turn those on or off. That's kind of a big deal. And so, we call this allosteric regulation. All right, so here's two different shapes of enzymes. We have our goldfish one we've been looking at, and we have this sort of four-leaf clover one we've been looking at. Allosteric enzymes have two sites. Okay, they have two places that things can bond to them. They have the regulatory site, which is different than where the substrate goes. So this is where the red square went that changed the shape. This four-leaf clover one here has sort of the spot in the little pits between the leaves of the clover, also has a site. So let's look at how this works. So we have molecules that bind to these allosteric sites. Okay, so we have molecules that bind to the allosteric site, and that changes the shape. And when you change the shape, function automatically goes with it. This can happen in two ways. You can have enzymes that are inactive until their activator shows up, or you can have enzymes that are active and then turned off. So these allosteric substances can either turn things off or on. So a key thing here is it could both inhibit or stimulate or turn on an enzyme. So it's not like it's one or the other, it's both. Let's take a look. Here we have our four-leaf clover shaped one. And you can kind of see that this, it's inactive. These little things are pinched close on the tips of these leaves of the clover. But once this teardrop thingy right here shows up, it bonds into this site right here. And when it does that, the shape of the active site opens up. And so right above me, you can kind of see that active site hole right there. And now that enzyme is ready to do its job. So this would be an enzyme you don't want to act until the activator shows up. So this one here doesn't turn on until the activator shows up. Now, we can look at this another way too. Okay, these are the same shape. So these are the same shape, but they're meant to be different enzymes. All right, now look at this one. It starts in the active form. Then, so if you look straight down here underneath me, that is where something would bond with this enzyme. It's active, it does its thing. If you look over here at this red thingy here, it jumps into sort of the spots between the leaves of this thing, and you can kind of see right here, it has, look at this right below me, it's closed off that little active site. Nothing can get in there now. So you can also have allosteric inhibition. This thing inhibits when it shows up. What's the big deal here? The big idea is allosteric things are switches for enzymes, and they can switch either on or off. And if you think about biochemical situations, there are sometimes the presence of something would mean let's turn it on. There are times where the absence of something would mean let's turn it on. Okay? And the opposite is also true. There are times where it's always on, and when something shows up, we better turn it off. So these things can sort of be feedback loops or controls to turn off enzymes, to turn on enzymes. If you are really interested in pharmacology, this is the kind of thing that can become very important if you want to look at drugs that turn these things off and on. And it can be enzymes, it can be protein pumps. A lot of times we are trying to block the action or stimulate the action of something through some sort of pharmaceutical, and this is one of many pharmaceutical mechanisms, okay? This isn't the only one, and a lot of them are way more complex than this. But if you think about all these metabolic pathways in your body, either inhibiting those pathways or enhancing those pathways is a real, that's pretty much how almost all drugs work. The problem sometimes is they also turn on things we don't want turned on or turn off things we don't want turned off, and we call those side effects. Okay. Sometimes we have what's called cooperative, see if I can pronounce that, cooperativity. That's totally wrong. But let's go ahead and look at it anyway. Cooperativity is when a substrate binds to one site and that stabilizes the active form of the enzyme. So what I want you to notice here is when this guy bonds here, it opens up the others. So when this purple jelly bean thing bonds, you can kind of see it changes, it opens up the other parts of the enzyme. So it bonds with one site and opens up others. It's still considered allosteric. All right, almost done here. So I've been kind of hinting at this the whole way, but we have what's called feedback inhibition. Okay, a feedback system is where when one thing goes up, the other one goes down. You've probably learned about positive and negative feedback, and that doesn't mean good feedback and bad feedback. It just means with negative, when something goes up, something else tries to get that back into balance. With positive feedback, which they aren't very common in biology, when one thing goes up, the other one goes up. When one thing goes up, the other one goes up. Blood clotting is an example of that. Breastfeeding is an example of that. Those are very, very rare in biology because they always end in a crash. All right, so feedback inhibition. This thing is an enzyme right here, this red little Pac-Man looking thing. And when we put an intermediate in here, it may do something to the next enzyme, okay? So starting with one enzyme may inhibit the next enzyme. And what we get is we get a feedback loop that keeps whatever product this enzyme is working on at a consistent level. And so this basically keeps biochemical processes in this feedback system where we don't get too much or not enough. Just like the furnace in your house. When the air conditioner turns on, the temperature drops until the thermostat says stop. Okay? We don't want to constantly be running the air conditioner. We just want it to run until it gets to the right spot. Same thing with the furnace. So feedback inhibition stops a metabolic pathway from going into overdrive. If the air conditioner got stuck on, well, that'd be really bad. If the furnace got stuck on, that would be really bad. So anytime these inhibition pathways break down, that's an opportunity for disease. All right, I'm going to go ahead and wrap this up here. The big key ideas are that enzymes don't always turn on. There are ways they can turn off and on, and the products of these pathways can be self-regulating.